Apoptosis is the process of biochemical events that lead to cell characteristic changes and death. There are two pathways of apoptosis. This is the first pathway, known as the extrinsic pathway. TNF and FASL is presented to the TNF and FAS receptor on the cell surface, which induces downstream signaling which activates FADD. Once the normal cell is presented to the TNF and FAS receptor, it is sentenced to apoptosis through the extrinsic pathway. This pathway involves the activation of caspases 8, 10, 3, and 7. These caspases destroy substrates of the normal cell, and the cell is then committed to apoptosis. Procaspase 8 is cleaved and caspase 8 is activated. This causes the activation of procaspase 3 and 7, leading to the activation of caspase 3 and 7. Activation of caspase 3 and 7 commits the cell to apoptosis. However, tumor cells have mechanisms of avoiding apoptosis. C-flip is an inhibitory protein that can bind to caspase 8 and 10 to form an apoptosis inhibitory complex. This prevents disformation and subsequently activation of the caspase cascade. Thus, the tumor cell is able to evade apoptosis. This is the second pathway, known as the intrinsic pathway. This pathway is triggered through cellular stress such as DNA damage. The intrinsic pathway is also known as the mitochondrial pathway. Upon cellular stress, the intrinsic pathway is initiated. The mitochondrial outer membrane becomes permeable, which is linked to caspase activation. Intrinsic apoptotic stimuli activate BCL2 proteins, which activate BAX and BAC, and increase mitochondrial outer membrane permeabilization. Tumor cells are able to evade apoptosis at this step of the pathway through the release of anti-apoptotic BCL2 proteins, which inhibit BAX and BAC. Upon mitochondrial permeabilization, a set of proteins including cytochrome C, SMAC, and OMI are released. These promote caspase activation and apoptosis. Tumor cells can also escape the intrinsic apoptosis pathway at this step. Inhibitors of apoptotic proteins can prevent the activation of caspase 9 on the apoptosome. However, SMAC and OMI inhibit IAP, which prevents tumor cells from escaping apoptosis. Cytochrome C binds with APAF1, creating the apoptosome, a protein structure which recruits and activates caspase 9. Tumor cells are able to leave the apoptosis pathway when Pro-T blocks apoptosome formation at this step. Caspase 9 cleaves and activates executioner caspases, caspase 3 and 7, leading to apoptosis.